Screen record. Screen record.
Screen record. Yeah, it's just an app that you can buy the app store. You can buy it. And you can do that on your computer. You can buy apps for your computer. Okay, guys. So there's been a lot of questions on the exams. The way they're not completely graded yet. Hopefully I'll have them posted tonight. We're very close to the final stretch. A few things. I ended up throwing out one question because it wasn't fair. Another question, there were actually two answers for, so if you got either of those two, you got right. And then there's another question that I'm considering whether or not I should throw out because it seems to be confusing for people. So we shall see. But so I was kind of disappointed in the exams. I think the average is going to be pretty much the same as I always have, which is somewhere in the 70s. But the difference is the distribution. Usually I have like people who are getting like between 20s and 40s, and then people who are getting like 80s to 110s. Whereas this time, everyone's kind of clustered between like a 65, 60, and like 100. And so it's kind of like in like a smaller space, I guess. I don't know. But it's very heavy on like the 70s and 80s, which isn't horrible. People aren't failing, but I like to see lots of A's. And I'm not seeing lots of A's. I'm seeing A's from people who I would expect to get A's. That's kind of my judgment. If the people who I'd expect to get A's get the A's, then I didn't totally screw up. If the people I'd expect to get A's are getting like B's and C's, then I probably screwed up the exam somehow. So it's not horrible, but I expected better. So we shall see what happens. But I mean, it should be good for you guys to know that no one like bombed like 20% in the exam. Right, that's good to know. No 20s yet. Well, I'm very close to the end. Anywho, that's where things stand. Okay, so they'll hopefully be posted tonight. And then I guess we'll draw the exam right up for those of you concerned. If you need to take the exam, I don't like people quitting. Quitters never win, or I don't know, something like that. I don't like people quitting. I'd almost rather you try to work with me and we try to work together to get through it than quit and take it again. Quit and take it again. It's like, that's no way to approach life, in my opinion. But anywho, so for those of you who are very concerned, let's try to work through this together. We'll all hold hands. It'll be fun. I wanted to get your thoughts about the online recitation. Who thought that was a good thing to have on an exam week? Okay. Who thought they would have much rather been in recitation because it was very hard for them to get through it on their own, and it's worth kind of having to deal with that on an exam week to kind of have the help of working with a group and having me there? Definitely a minority. Now, I have to say, I was watching you, I tracked who actually read the paper. And I'm very disappointed that a lot of people never even opened the paper. Probably no one in this room, because you guys are all the good students. But those of you at home, I saw you, and I know exactly who. I can try it. So it is a little disconcerting when people don't even open the paper. Now, I don't normally track it to recitation because I can tell when I'm standing there with you. But I don't know. So I'm going to actually take a look at the grades and see if it reflects that or if we have a cheating problem. So that's going to determine whether or not I try this again. But it looks like overall you guys enjoyed having the extra time to study and that it was mostly worth it. And for those of you who needed the extra help, come see me next time if we do this again. You have a question?
Was there a mistake in the recitation quiz?
Was there a mistake? There was a mistake on the recitation quiz, and it was, did you see, I posted an announcement about it. There was a mistake, and it was regraded. I believe it was regraded. Yeah.
You said like omit. So do you mean that group question when you say that?
Yeah. I think so. There were two mistakes recently. Oh, right. I had to get rid of the question because there was a typo in the answers. And so there's no way to fix that after once I just kicked out the question. Yeah. There was also a very minor error on this weekend's quiz, but whoever told me about it right away, like on Saturday. So I fixed it probably before most of you saw it. You saw? Were you the one who emailed me? No, but you saw it. But it got fixed. Okay. Anywho, let's go. Oh,
What
stump.
question was it?
The matching one. Okay. Stump the teacher. We had a stump the teacher where I couldn't, for the life of me, remember TF2H. Those of you who sent me TF2H will get a point. Those of you who just told me DNA, RNA polymerase opens. No. We're talking about eukaryotic cells there. So that's that. For those of you who are worried about your grades, get all these stump the teachers. Sometimes I don't get anything from people. There's always some people, but I think some people have at least 3 or 4 extra credit points from doing these repeatedly. Okay, so far we just kind of started with the transcription basics. DNA versus RNA, DNA polymerase versus RNA polymerase, kind of comparisons, just general thoughts and ideas about transcription. And so this is where we ended. So this is a process of transcription in bacteria. We dive right in. Right, so you've got something called a sigma factor and RNA polymerase. Those are your two main components of the process, that plus obviously the DNA. And so the whole point of your sigma factor is that it binds to the DNA. Where? At the promoter. Right, so that's a big part. And it helps to direct RNA polymerase to the right place to start transcription. So your sigma factor will bind, and then RNA polymerase will bind, and then you'll get melting and opening up of your DNA, and then you'll have this abortive initiation where you'll get these short transcripts, which you guys are reading about in this week's overly short paper. So we'll learn all about abortive transcripts and recitation this week. And after a little while of making these short transcripts, then the sigma factor is going to detach, and RNA polymerase will leave the promoter and go through elongation of your transcript until it hits a terminator, and then it will terminate transcription and release the RNA. Okay? Yeah. RNA polymerase. Did I say DNA polymerase? Yeah, you never know. You never know what I'm going to say. RNA polymerase, we're talking about transcription. If I ever say DNA polymerase, throw something at me. Well, don't really throw anything at me because it could injure me, but just mentally throw something at me. Okay. So how does RNA polymerase know when to start and stop transcription? What kinds of elements in the DNA are present for starting transcription? And a DNA element, so it's a sequence within the DNA that tells you don't give me the exact sequence, but maybe a name. Promoter, right. The promoter is the sequence in the DNA that says to RNA polymerase, come here, start transcription here. Okay, and so how in bacteria, we just talked about how there's a sigma factor, and that's what the sigma factor binds to. It binds to the promoter. And so that's what directs RNA polymerase to the transcription start site. And then if you saw here, you have this termination signal, and that creates this stem loop structure in the RNA and results in release of RNA polymerase and the RNA and termination of transcription. Yes? No, so it's actually the structure, So what's happening is so what tells it to stop transcription, right? That's what you're asking. So this sigma factor is gone. It's long gone. And that's really just about directing RNA to the transcription start site. But what happens is you'll get this transcription, that blue line here is your RNA coming out, and then you'll get to this certain sequence that's going to base pair and form this hairpin structure that you see right here. That's your RNA in the hairpin structure, and that's going to signal for termination. And then that RNA is going to be released, and the RNA polymerase is going to release from the DNA. And so we'll go a little bit more into that. Any questions about this general process? It's actually fairly simple, because you just need to remember a few main points. One is, how does RNA polymerase get to that gene that's going to be transcribed? That's the promoter. That's a sigma factor directing RNA polymerase to the promoter. And then how does termination happen? In between, all you have is elongation, right? That's the extension of your RNA. Okay, so the sigma factor is recognizing a specific DNA sequence at the start of the transcriptional unit, and that's the promoter. And so you can see the two of them together are what recognizes the promoter. So the sigma factor is actually directing RNA polymerase to the promoter. And there are different sigma factors, so it's not one thing. And those different sigma factors recognize different promoters. And so you should automatically think to yourself, oh, this could be a point of regulation, right? Different sigma factors recognize different promoters, so they're going to direct RNA polymerase to different genes. So there are primary sigma factors that bind to promoters of essential genes. So these primary sigma factors are plentiful and are involved with transcription of a lot of different genes. But remember, if you control somehow the levels of these promoters, you're controlling transcription. So that's a point of those genes. So that's a point of regulation. Then there are alternative sigma factors, and they are binding to genes whose expression is regulated. So for example, heat shock proteins. So so remember, you have constitutive expression of genes. What does constitutive expression mean? Constant. Yep, sounds the same. Easy to remember. So constant expression is just a gene that's always being expressed. These are like little more housekeeping genes required for just general cell metabolism, growth, stuff like that. And then there are inducible genes that get induced in response to certain environment, so for example, heat shock proteins. Heat shock proteins are induced by heat shock. Okay? And so there are alternative sigma factors that are going to be upregulated and active under certain conditions and induce expression of certain genes. Okay? So the core RNA polymerase plus the sigma factor is known as the RNA polymerase holoenzyme. So only together, sigma factor plus RNA polymerase, can you actually bind to the RNA and have gene expression. Okay? So what this figure here is showing is you've got your DNA, and then you've got these different elements in the DNA. By elements, I mean sequences within the DNA that are recognized specifically by the sigma factor or components of RNA polymerase.
Is that in bacteria? That's in bacteria.
Huh?
Is that only in bacteria?
This is all bacteria. Sigma factors, bacteria. The analogous system in eukaryotic cells are your general transcription factors and gene regulatory proteins that we'll talk about. Yep, so this is all, so we're going to start simple with bacteria. Good question. Good clarification, I should say. Okay, so sigma factors, these are proteins, right? And they contain different domains with different functions. You guys remember what domains are? Parts of proteins that do specific things. So they contain different domains with different functions. So this right here is showing the sigma factor, which is this big globular structure. around binding to DNA. And so you can probably see it more closely if you're looking on the PowerPoint, but it's here in the middle. There's the helix. And it's showing that there's this sigma 2 domain that binds to the -10 promoter element. So this is a sequence that's 10 base pairs downstream from the promoter. So +1 means that's where transcription is going to start. And so your promoter is usually before your transcriptional start site. This is actually one of those things that totally eluded me until graduate school. Because when I took mobile bio, I just remember someone being like, +1, -10, -35, blah, 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 blah. And it was just like number soup. It might be simple to you, but I'm going to like try to spell it out because it confused me for some reason a long time ago. But it really just means that you've got your start site, and then all these proteins are going to be recruited just prior to the start site. And they recognize certain sequences in the DNA. Okay? And so this sigma 2 domain is going to bind to the -10 promoter element, and that's involved with opening up the DNA. Then there's this sigma 3 domain, so it's still part of sigma factor. The sigma 3 domain, it's going to bind to an extended -10 if it's present. So there's this -10, which is here in green, and then there's this kind of yellowish, which is just this extended bit of -10 element. And then there's this sigma 4 domain up here that's going to bind to the -35 promoter element. So you can kind of imagine how it looks. So you look at the arrow. This black part of your DNA is just sequence, but this green part is your -35 bound to the sigma 4 element, and this green part over here is your -10 that's bound to your sigma 2 and sigma 3 domains. Okay, main point, sigma factor bind to DNA. So this holoenzyme, that sigma factor plus RNA polymerase, it's going to bind weakly to DNA except for at the promoter. And that's where it's going to bind tightly. It's going to bind strongly. And this is just showing your sigma factor and your RNA polymerase. So you can regulate your sigma factors, right? So if your sigma factor is available for binding to DNA, then you can bind to DNA and it can recruit RNA polymerase. If it's for some reason not available, or if the DNA is not available, then you can't recruit RNA polymerase. So when you're talking about transcription and regulation of transcription, the question you should always ask yourself is, can RNA polymerase bind, right? And there are many reasons that it can or can't. So for example, there are these pro-sigma factors, and they contain, like any pro-protein, they contain some kind of part that needs to be cleaved off. In this case, they contain an inhibitory domain that needs to be cleaved before it can associate to RNA polymerase. So that's regulation there, right? Sigma factor can only actually function to allow RNA polymerase to bind to the DNA when the pro-sigma factor's been cleaved. And then there are anti-sigma factors, and these are proteins that bind to sigma factors and inhibit their function. So they inhibit the ability to bind RNA polymerase, bind DNA. Okay? So, do you guys understand why you might want to regulate your sigma factors if you're a bacteria? Should all genes be transcribed all the time? No. So this is one mechanism for regulating. You can have these pro-sigma factors, and until you would regulate activity of the enzyme that cleaves them. You can have these anti-sigma factors. You can upregulate those when you want to inhibit function of certain sigma factors, okay? And therefore expression of certain genes. Questions about this concept? Yes.
So can you like inactivate sigma by phosphorylating it or any other way other than just removing that inhibitor?
I'm sure there are other ways. So the question was, can you regulate sigma factors through post-translational modification, other than just these mechanisms? I'm sure you can. I don't know exact mechanisms off the top of my head, but I'm sure you can. Yes?
Is there also because like sometimes bacteria they're small organisms and like a genus pretty small compared to like other bigger organisms. Would you want like any type of sigma factors to be highly regulated so like they wouldn't like it would like conserve energy in like turning on and off the genes? Like you wouldn't want you to spend too much energy on.
So you would want to regulate sigma factors to conserve energy. You mean in terms of rather than constantly making new sigma factors, just have these like anti-sigma factors, for example? Maybe. I could see it as being conserving. Yep.
What's the difference between the anti and the pro? Because they both inhibit.
Right, they're both inhibitory, just different mechanisms. The difference between the pro-sigma factor and the anti-sigma factor, pro-sigma factor is a pro-protein. Any pro-protein is a protein that's not active until some kind of part of it is cleaved off. You can if you wanted to visualize it in some way, you could think of it as a protein that maybe folds over and binds some active site, and then you would cleave that part that folds over off to activate it. Okay? That's just a way to visualize it. It's by no means an exact mechanism, but that's one way to think about it. Anti-sigma factors, they're just inhibitors of sigma factors, so they're going to bind to and inhibit sigma factors. In both cases, they're inhibitory. Any other questions? Okay. So just to go through bit by bit. So first you need to open up the DNA. In bacteria, there is no actual helicase. The activity is within RNA polymerase. So the sigma factor is going to bind, and it's going to bring in RNA polymerase, and then you're going to open up your DNA. And then you get this inefficient transcription of the first 10 or so, 10 to 15 nucleotides, and that's called abortive initiation. And so then you get, so that's what that's showing right there. And so then you get the release of the sigma factor, and then you get promoter clearance, meaning you clear the promoter, you go past the promoter, and then you go into elongation, and then you get the elongated transcript. Yes?
Why is there
Why is there abortive transcription? So in the paper that we're reading this week, it's all about abortive transcription. And I don't know why it happens. I still am not satisfied. It appears to be all RNA polymerases do it. But I don't know how much evidence there is for this in eukaryotic cells. It's something that definitely bacteria do in vivo. There's got to be a reason for it, because why would it? It seems very wasteful. And there are theories about, well, maybe these small transcripts do other things, but I don't know what they are. Yeah. I actually pulled up a review today to read about it because I was so unsatisfied. It's a new paper that we're doing in recitation this week, and it's very short and sweet and simple, but like it opened up so many questions for me, and I pulled up this review to read about it, and then I'm completely unsatisfied with like the whole purpose of abortive initiation, because it happens. There's evidence that it happens, but why? Like I feel like it's so inefficient. So why has E. coli not evolved a more efficient RNA polymerase that doesn't do that? So there must be some advantage, I'm guessing. Okay, other questions? It's like a party up here. Okay, so then you get rapid chain elongation until the termination signal, which looks like a hairpin. So you form this hairpin structure, and then the polymerase stops and releases DNA and RNA. That's your termination signal. So there's a consensus sequence for a major class of E. coli promoters. Do you guys know what a consensus sequence is? How about this? What does it mean when a group has consensus? Everyone agrees. So what consensus sequence is a sequence that is agreed upon by all the promoters. Sorry, I cracked myself up. It's true, you know. That's how you know you're a complete science geek. It's like when you're like cracking yourself up and everyone else is just like. So there are consensus sequences for a lot of things, like different domains have consensus sequences. That means that in all these different proteins that share this domain, they have this sequence that's the same among all the domains. And so for promoters, there's a consensus sequence for one class of E. coli promoters, one major class of E. coli promoters, and it looks like this. So remember here you've got your -10 and your -35 again. So that means that your transcription start date should be over here. That would be your +1 over here. And this is where, what's going to bind to this -10 and -35? Your sigma factor, right. And so this is a consensus sequence, which means most of the time you find a T here, most of the time you find an A here, and so on. So the strength of binding to sigma factors is involved in the frequency of transcription of a particular gene. So if this consensus sequence binds strongly to a sigma factor, then you're going to get transcription of that gene. Kind of makes sense since your sigma factor brings your RNA polymerase in, right? Okay, so here's a practice problem. So you're analyzing a newly sequenced bacterial genome. You guys passed my class, you went on to graduate school. Here's your project. Your boss gives you a bacterial genome and says, I need you to identify all the transcriptional units in this genome. And you're like, that seems like a lot of work. And so the genome's about 5 megabases. And so that's huge. And you decide it's time to use your bioinformatics training to write a computer program to identify transcriptional units. Propose a logical way to identify all the transcriptional units in the genome. So I want you to think about what we spoke about just now, about consensus sequences, about what they are and about what they do, and think about how that information can be used to look at a whole bacterial genome and identify every or all or most transcriptional units. And what's a transcriptional unit? That's kind of promoter plus gene. Okay? So go ahead, talk to your friends for a little bit. I will circulate. I will make sure everyone's on Facebook, updating their status. Now, cool class. Practice problem. People post the weirdest stuff. I've decided I need to kill my Facebook. And just like, because doesn't it like just piss you off sometimes when people are like, great dinner. Maybe you guys are cooler because you're younger, but when you get like older and have a family and all your friends have families, it seriously is like date night. Like it's like, you're like awesome. I'm telling you, you're going out. Okay, sorry, I totally went on a tangent. So you guys go and talk about identifying your transcriptional units and what kind of computer program you would write, and I will come and help you. You don't need to actually write a computer program. Who's complaining? Don't sit and write a computer program. If you can, you said you're doing bioinformatics, so you can write a computer program. But no one else is required, only Shira is required to write a computer program right now. But right, so just, I want you to think about what you would do.
I was thinking we figure out. That's kind of
What did they show?
I mean, that's what I think it is.
Yeah, I didn't write it.
Or you could come up with it. Like lines and flows. You can do that. Go back on. So starting from your point, so more routine in the beginning, right? Less. No, I think so the thing is, you have more teeth in the beginning, right? Minus 35, that's good. And then as you move, I just skip those 15 to 19, you decide. Just look at it, so blue is A, right? So and then you move to that, again, when you start that minus 10 part, you have 14. As you go towards the end of the year. So if you just.
Okay, guys. So what did, I heard some interesting ideas. So what did we decide? So you magically now have a program, and you're writing a computer program, and you're trying to identify all the transcriptional units in this genome. So I got a question about, well, what are you starting with? You're starting with your whole sequence of your bacterial genome, all the bases. You've got them all ready. You've got the sequence. It's in a computer program, and you just need to mine it by telling it to look for something. And so, what would you tell it to look for? Promoter. Specifically, what sequence? Basically, this sequence, right, this consensus sequence, that's why it's useful. Because you know when you find this minus 10 and this minus 35, then you know that sigma factor is going to bind there, and you know that that's a likely place where RNA polymerase is going to be recruited. Now, mind you, this is a consensus sequence for some E. coli promoter, so you would have to identify consensus sequences for multiple different types of promoters. But this is a starting point. So so you know where it starts. What would you look for defining, because transcriptional unit is promoter plus the rest of the gene, the coding sequence, right? What would you look for to know what the end is of your transcriptional unit? AT rich region. What's at the AT rich region?
So that's where you can see the consensus.
But so this is in your promoter. But what about the end of your transcriptional unit? Terminator. Okay, terminator, so that's one. So assuming that the termination signal has a consensus sequence. What else? Did I hear another thing? AUG.
I just have a question. Is there possibly a way for, if since you know that there's a hairpin, created at the end, is it possible to, like, tag a hairpin?
So what do you mean by tag it? Because this is just a text file you're working with on your computer. Right? So you're given the whole genome sequenced already. So you have a whole bunch of bases in the order of the genome. So right now you're asking me how to identify the termination site, right? That's really your question. And so how would you identify the termination site? So if there's a consensus sequence for termination, you can look for that. And so you said AUG. Why did you say AUG?
Because that's usually the end.
So what you really mean is
sorry.
TAA or
No, the UG, sorry.
so what you really mean is stop codons, right? So looking for stop codons. So that we'll find at the end of the protein, right? Okay. But maybe you want also some untranslated regions, so looking for termination signal. That's helpful too. But these are all different things you can look for. Yes.
Don't you want to have like GC regions for innovation between bonds versus two?
Would you want to have GC-rich regions for termination? So what's your thinking?
Because 3 bonds versus 2 bonds, you can stop in one.
Okay, so your thinking is a termination signal might stop opening the DNA, so maybe it would be GC-rich. I don't know. Well, so the termination signal actually is transcribed, and so you actually get RNA there, and it forms this hairpin loop. And that results in RNA polymerase coming off and releasing from the DNA and releasing the RNA. I'm not sure, though, if there's anything beyond that in the DNA. It's a logical reasoning process there. Any other thoughts? We pretty much got it covered. Okay. Termination signals form a hairpin. So what do you think they might look like sequence-wise in the DNA or in the RNA? What do you think, like, if you had to write a DNA or actually RNA sequence that's going to form a hairpin, what do you think it might look like?
It would be like Fiji, Omni, and it's not Fiji.
Right,
A lot
right.
of repeats, so they can actually.
Right, so you'd have some kind of inverted repeats probably, right? Let's see if I could do this without confusing myself. Right, so really what you end up having right, that's your hairpin. And so you're going to have base pairing between all of these bases, which means if you have a U, C, G, U, then on this side you would have to have A, C, G, A, U, A, right?
And then this part would probably not have any base pairing at all, which is why it's a stem loop. So that's pretty much what it would look like, except for it would be straight, and it would be like A, C, G, A, U, A, and then dot, 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 and then it would be U, A, U, C, G, U. Oops, there we go. Right? Does that make sense to people? Yeah.
Doesn't that mean for every single transcribed RNA, they all have to have some sort of complementary unit right there? They all have to be easier.
I can't say for every single, but this is a common mechanism for termination, is that you would have this sequence that would allow for formation of the stem loop. Yes.
So even the mRNA that has a 5' top and they will have that structure first. How do we transfer out of the nucleus with that structure?
It does. Yeah. So your question is,
That strengthens the transfer.
well, is it? So you're saying to yourself, wait a minute, if it's got the stem loop, how's it getting out of the nucleus? Well, that isn't straight when it's coming out of the nucleus.
It's not.
Is it straight? Yay, let's see. Yeah, so actually it's bound by all these proteins, and it's in a circle actually. So so we'll get there, but you're getting all this processing, this capping, the polydenylation, and then you've got like a cap binding protein, and you've got all these different proteins that actually bind to the RNA and kind of bring it into a circle shape, and then it's exported out. So we'll see. So something like this is not a big deal really, but I'm guessing it's going to be cleaved.
For the microRNAs.
Well, so that's a different microRNA is another story. Yeah. So this is the first instance of an RNA secondary structure playing a role in gene expression. So just kind of keep in your mind that we're going to see a few times some kind of RNA secondary structure playing a role in gene expression. And it's nice to, when you're learning about things, group things into categories, because then you can start to think to yourself, oh, well, we see it also with splicing, we see it also with silencing, we see it with these different situations, all different, but you're having specific structure of RNA play a role in gene expression. Okay? Yes?
So for the DNA sequence, wherever we had the U, we did T, right?
For a DNA sequence, wherever you have the U is a T, yes. Yes. Okay, so transcription can occur using different strands of the DNA as a template. So remember, DNA is double-stranded. Either one could be your coding strand. Okay? It just depends on how RNA polymerase is brought in, and that depends on the orientation of that -10 and -35 sequence, that consensus sequence. That's going to dictate how your sigma factor binds and how your RNA polymerase comes in, and the orientation that transcription is going to go in. So if you think about it, that gives a huge amount of flexibility, right, to the genome. A lot of genes that can be included. Right? So in bacteria, the gene orientation is determined by the orientation of the promoter and how sigma factor binds to it. Right? So you'll have your double helix, and you can either you can have an RNA polymerase that's moving from left to right using the bottom strand as a template, or you can have an RNA polymerase that's moving from right to left and using the top strand as a template. It just has to do with where your promoter is in the orientation that it's in. Okay, so that's bacteria. So let's talk about transcription in eukaryotic cells. It's a little bit more involved. So with initiation, you need the assembly of what are called general transcription factors. These are all your TF2 and letters, right? That you guys had to do matching with on the codes. And they all have a different function. And so the role of these general transcription factors is to bring in RNA polymerase. And then also phosphorylate RNA polymerase's C-terminal domain. And we'll talk more about that. Right, so the first thing that happens is the same as bacteria. So as we're going through this, I want you to be kind of comparing and contrasting in your mind bacteria versus eukaryotic cells. And you'll see the theme is the same. Promoter recognized by something brings in RNA polymerase, essentially, right? It's just a little bit more, there's a little bit more involved. So first you're gonna recognize your promoter, which is called the Tata box, because there's a lot of T's and A's. And that's recognized by TBP, which is Tata binding protein. So you shouldn't forget that, because Tata binding protein binds to the Tata box. And that's a subunit of TF2D. So that's your first step. And then TF2B comes in and it binds. And it's involved with the accurate positioning of RNA polymerase at the transcriptional start site. So again, this is very much involved in orientation of RNA polymerase. So when Tata binding protein binds to the Tata box, it actually distorts the DNA. And so this is believed to serve as a landmark for assembly of other general transcription factors. Because you got to imagine, if you've got DNA and there's a promoter, there's got to be some way for all those transcription factors to get to the same place, right? If you think about it, like it's just like DNA is huge. And just because you have Tata binding protein there doesn't mean that all the other general transcription factors are going to get to the same place. And so it's believed that this distortion of the DNA kind of creates a landmark for assembly of these other transcription factors. And so this is showing Tata binding protein binding to DNA. You can see the DNA is bent, right? Yeah. The what? So this is the distortion is a direct result of Tata binding protein, which is a subunit of TF2D binding to Tata box in the promoter. Next. Yeah, so you can kind of think of like you've got like hikers climbing to the top of a mountain and they're going to put their flag in the top and say, I am here, on top of this mountain. And then all the other transcription factors are like, let's go find the flag and go there. I don't know. It's like a mountain. Yeah? Okay, we'll work on that. We'll work on my analogies. So just like in bacteria, there are all these different DNA elements that are found at RNA pol 2 start sites, and these are all specific to RNA polymerase 2. Remember, RNA polymerase 2 is involved generally with transcription of what? mRNA. Something's going to become a protein at some point. And that paper we did last week, also we learned that it's involved with transcription of microRNAs, right? These DNA elements are found at RNA pol 2 start sites specifically. So you've got this Bri element, this Tata box, this INR element. This is your +1, your transcription start site. And this chart tells you what binds where, and it gives you consensus sequence. So again, if you're a bioinformaticist working with some new eukaryotic genome, this could be useful, maybe. If you're trying to figure out where transcriptional units begin and what binds where. So this TF2B binds to this Bri element, Tata binding protein binds to the Tata box. INR, TF2D binds there. And then there's this also +30 region, which TF2D binds to as well. So, do you think these elements, these DNA elements and the transcription factors are involved in regulation of gene expression? How do you think they're involved in regulation? When I say regulation, I mean whether it turns on and off. How do you think these can be controlled in such a way that you're going to get gene expression or not?
Could you have like an inhibitor bind to the spot?
Yeah, you can have an inhibitor bind to the spot so that the transcription factors can't bind. If you have a protein binding to your TATA box, then your TATA binding protein will never come. And then that's kind of your first step in the whole process. Well, whatever. So, yeah. So controlling whether or not your general transcription factors can assemble at your promoter, it's a huge point of regulation. Question? Huh? Okay, go. You can degrade transcription factors. So by controlling the availability of transcription factors, you control whether or not transcription is going to happen. Right, and so these are all obvious answers, right? Either the binding site is there or it's not, right? Because maybe it's covered up, or what the binding is there or not, right? So then I told you there are all these consensus sequences. How can they be useful in identifying transcriptional units? This is just an extension of the question we just did. If you look for these different consensus sequences, then you can start to identify. You know your Breeze element is minus 35 and your TATA is minus 30, and you know the spacing. There's general spacing in the promoter where you should find these consensus sequences. So you can start to identify transcriptional units this way. What kind of genomes would you expect to find these consensus sequences in? Would you find it in an E. coli genome? Would you find it in a yeast genome? Yeah, so they're eukaryotic promoters. So you would find them in eukaryotic genomes. So remember, always be comparing in your mind prokaryotic, eukaryotic, or bacteria. I always get very nervous about just grouping all prokaryotic together. Yes?
We're wondering what the backslash is between
Ah, thank you. So with consensus sequences, there are different ways of showing it. When it says G/C, that means a G or a C can be there. So this is G or C, G or C, G or A, and then C, G, C, C. And N means any amino acid. Any nucleotide. Oh my lord. Right. Yes.
Why did you say it would be
Because these are this consensus sequence. This whole system is only eukaryotic. Prokaryotic is sigma factor binds to that one sequence, that one other consensus sequence that we saw, recruits RNA polymerase. This is eukaryotic, where you've got your TATA box, got your Brie, got your RNA, your DB. That's all specific to eukaryotic transcriptional units. TF2s, all eukaryotic. Okay. Okay, so you've got your TATA box, you're bound by your TF2, let's see, where are we? Right, your TATA box bound by your TF2D, and then your TF2B comes in, and that helps to orient RNA polymerase. And then this allows the rest of your general transcription factors to bind, and then that brings in RNA polymerase 2. So you've got TF2E, TF2F, TF2E, TF2H. TF2H is the one that I forgot about, and that you should be throwing tomatoes at me for. Actually, you just get a point for my bad memory. It has helicase activity, and it also is involved, well, it plays a role in the RNA polymerase CTD, C-terminal domain phosphorylation. Okay, so what happens is helicase activity, opening up the DNA for transcription to begin,
And then C terminal domain, that's that little tail coming out with the phosphate groups on it. That TF2H phosphorylates the C terminal domain, and you'll see that that's really important in processing, activation and processing. So based on what I just told you, yes, C terminal domain of RNA polymerase. So it's a part of RNA polymerase at its C terminus. So based on what I just told you, what domains do you think TF2H might have? What types of domains? Yeah. A kinase domain, right? Because it phosphorylates things. What else? Yep. A bromodomain. Maybe. That wasn't what I was thinking of, but now you're like, let me just throw everything in. You know, based on the fact that, I don't know. So if you're associating gene expression and acetylation, I can see how you would arrive at bromodomain. I'm not sure though. We should look that one up. But what else? What other domain do you think, based on what I just told you, TF2H is going to have? Yeah.
Maybe some sort of a removal proteins that will remove your general transcription factor. Is that what are you?
Nope. Proteins that remove general transcription factor. I mean, that does disassemble. That does. But I told you guys it had helicase activity. Yes. Some kind of helicase domain, and then that goes back to what you said. ATP binding probably does have ATPase domain. Probably. Okay, good. Using information from different places, bringing it all together. How are we doing? Yes.
Would the CF2H need loading proteins too, or
Would it need what?
loading proteins?
Loading protein? Oh, like helicase loader for DNA synthesis. Is that what you're thinking? Well, so TF2H you can almost think of as kind of loaded in a way by the binding of these other general transcription factors. They create the binding sites that allows for binding of TF2H. But it's not, so it has something that's going to recruit it. Just like in DNA replication, there's CDT1 and CDC6 that help to load helicase. Yeah.
If you, this picture, why is the TF2 sticks onto the polymerase and then sticks, so that's not even on the DNA itself in order to be activated?
Right. It binds to polymerase. There's a whole bunch of other factors that bind to the polymerase.
But then they won't
The TF2F is the one that they talk about in the textbook. Yeah. Yeah. So it's like, it's not as simple as sigma factor binds promoter brings RNA polymerase. That's how we start simple. Here, there's a whole lot of other factors involved in recruiting and activating RNA polymerase 2. Okay? Okay, so this is just a list. It's of what we just went through. Some people like lists, other people like pictures of the different transcription factors and what they do and what they bind to and how they're similar to the bacterial factor, right? So you have TF2D, which is TATA binding protein, TAFs, but basically TF2D. And TATA binding protein binds TATA box, and TAFs, which is also part of this, recognize other elements in the promoter. And so it's analogous to sigma factor domains 2 and 4. Remember we looked at that, so you can kind of go back and take a look at that. TF2B recognizes debris element and establishes correct orientation. This is analogous to sigma factor domain 3.2. TF2A further stabilizes TATA binding protein TF2B binding, so it's kind of about bringing in all these proteins. And this is analogous to a subunit of the bacterial RNA polymerase. Then there's RNA pol2 and TF2F. And so TF2F, which I didn't talk too much about, that's the one that binds here to RNA polymerase. TF2F suppresses nonspecific DNA binding of RNA polymerase. You don't want nonspecific binding of RNA polymerase, right? Because that needs to be very specific. You want to transcribe the gene you want to transcribe. You don't want to bind them in some other random place, right? And so that's analogous to the alpha subunit of bacterial RNA polymerase. No, that's analogous to RNA polymerase in bacteria. And then TF2E only recruits TF2F, and then TF2H, and TF2H is your helicase activity. And there's no analogous factors in bacteria for those two. Okay? So it sounds like a lot. This is one of those things where you just need to find a way to know it. TF2H, H for helicase, so that's an easy one. TF2D, if you remember, is how to binding protein that's part of that. Then you'll remember that that's the first step to recognize Tata box. Yes?
If TF2H deals with helicase, why would it be on my RNA, like, why would it stick to my RNA polymerase when I'm trying to unwind DNA? Like, I feel like.
Did you need to unwind your DNA to do transcription? What do you think? Right, because your DNA is like your template for transcription. So you need to get in there open because, and as you'll see, as we start to go through the actual mechanism of transcription, you're going to have those bases coming in and base pairing with your template strand.
Why
Your
would it
DNA.
just attach to like your actual DNA without your RNA polymerase? Like if I'm trying to unwind my DNA, why not just attach to the DNA and move along and unwind it before my RNA polymerase comes in?
Okay, so I see what you're saying. So you're wondering why it's part of a complex rather than acting independently to open up your DNA.
Yeah, before I attach my RNA.
Kind of like how, well, it's kind of similar. So let's compare it to DNA polymerase and the helicase there. It's all part of one complex, right? And so it's ensuring that you're opening the DNA right where your polymerase is acting. Whereas this is ensuring that you're opening your DNA where your polymerase is acting. What do you think is going to happen if you open your DNA and there's nothing there? It'll probably just close again, right? Because it wants the base pair. I mean, I see your reasoning because you're thinking about DNA polymerase, right? Yeah, but it makes a lot of sense to have something right there where your active site of your RNA polymerase is to open the DNA right there. And it's actually just a little bit that's open. Large enough space for a few base pairs. Yes? The BRE, that's just this element that is in the promoter region. It's a DNA element. Yes?
So if, this might be a general question, but if one of the proteins is missing, you won't be able to go forward, would you?
If one of the proteins is missing, will you be able to do transcription? Is that your question? I'm guessing you'll have a hard time depending on what that protein is, unless some of these more minor factors that I don't talk too much about. I don't know. Maybe there's some other factors that have redundancies. But my guess is without these factors that I spoke about, that job won't be done. So clearly TF2H and TF2E are going to be a problem. TF2D, you won't get recruitment to the proper locations. You can kind of go one by one through each factor, think about its function, and then think about what wouldn't happen if it didn't work, right? And that's a really good way to study because it helps you to think very deeply about the functions of things, right? Yeah.
I know that DNA replication and transcription is two different things, but I think it's something I learned somewhere that they might occur simultaneously when you have that replication bubble and your DNA replication complex are running both ways, and at some point RNA polymerase might bind it somewhere to that one strand of DNA and start
So your question is,
transcribing.
can you have transcription within the replication bubble? Maybe. I don't see why not, but I also know during S-phase transcription is fairly low. I don't see why. I wouldn't say it doesn't happen.
But after S-phase, you have the G-phase when you have protein synthesis and grow size, so you might want to have that RNA.
Right. So yeah. So and there are different genes that are expressed in different phases of the cell cycle because you're going to need different genes to do different proteins to do different things. Right. So it's very possible that you might have transcription of certain things, but then don't forget that your origins. There's so many origins and so many places, and so like where I don't know what regulates whether or not you're going to get transcription within that bubble or whether it's going to happen. I also don't know. I mean, when you open that DNA for replication, it's single-stranded for a very short period of time. You get single-strand binding proteins there, and then it very quickly becomes double-stranded. So it's not, I don't know. Yeah, something to think about. How are we doing? Are we happy? It's Tuesday. Got about 5 minutes left. Okay. So there are additional proteins involved. Are you happy to hear that? So two complexes of proteins that are involved to keep in mind. One is mediator, and it's a complex of more than 20 proteins required for transcriptional regulation, and we will get to that in more detail. And then, of course, your histone modifying and nucleosome remodeling enzymes, right? What are your histone? Wait a minute. Why would you have histone modifying and nucleosome remodeling enzymes involved with transcription? Right, okay, good. So you guys are like, duh. But I was just making sure that you guys are compartmentalizing. You know, people like to be like, we're done with that. But no, it's for real. That was stuff to keep in your brain because it relates to that. Right. So all that acetylation stuff that we talked about and the recruitment of chromatin remodeling complexes to open up the DNA for gene expression. This is what we were talking about. That makes this DNA available for all these different transcription factors to come in, recruit RNA polymerase, and activate gene expression. So take a minute to tie it all together. Tell me when you're done. Okay, so mediator, right? So transcriptional activation requires binding of specific transcriptional activators or transcription factors, right? I always say to you, oh, and there's some signal from the outside, and there's a receptor, and there's signaling, this thing goes into the nucleus, that's called the transcription factor, and it binds to the DNA and activates gene expression. Magic, right? So all that happens. So these are transcriptional activators usually that I'm talking about. Sometimes people call them transcription factors. And so they will bind to these enhancer regions. So these are these DNA elements, so it's DNA sequence that's far from the promoter and the transcription start site, far away. And it will actually function to recruit general transcription factors and RNA pol2 to assemble at the start site. And so you can say to yourself, well, how does that happen? You're saying it's far away. Well, what really happens is this mediator complex plays a role, and you get this looping of your DNA. And so you'll get your transcription factor activator protein binding to your enhancer, and then you have this assembly of your transcription factors, and mediator is this protein complex that's allowing that communication between this distant enhancer site where your transcription factor is binding and then assembly of your general transcription factors and recruitment of polymerase and histone modifying enzymes and chromatin remodeling complexes, and it kind of all comes together, and then you have transcription. So it helps the distant transcriptional activators communicate with general transcription factors and recruit RNAs. I always like this picture because I feel like it ties everything together nicely. Questions about this? I think this is where I might stop. Yep.
Upstream of the motor, you have double-stranded DNA, right? Because you don't have helicase activity.
Upstream of your promoter, you have double-stranded DNA. You have double-stranded DNA everywhere here until your TF2H comes in and starts opening. Yep.
A lot of proteins in a lot of ways to cause inhibition, right? To inhibit. There's a lot of ways to inhibit.
There's a lot of ways that things can be inhibited. And so we're the repressors can bind anywhere along that.
Even like the TF2D, if something bounds like a TF2D, preventing it from binding to the plasma body. That can also
Anything that prevents recruitment or
I didn't do it during my time.
Exactly.
That's what they're talking about.
I was like, I don't know.